Hi everyone, so our topic for today is about transcription and translation. The differences between the transcription and translation. So first, let me define or differentiate the two. First is transcription. Transcription is the process where the genetic information on a DNA strand is transferred into an RNA strand by a series of polymerization reactions catalyzed by enzymes called DNA-dependent RNA polymerases. So, transcription is the first of the several steps of DNA-based gene expression in which a particular segment of DNA is copied into RNA. In transcription, the DNA sequence of a gene is transcribed or copied out to make an RNA, while translation is the process of protein synthesis where the information of RNA is expressed in the form of polypeptide chains. In translation, it is the process of, from the word translating the sequence of messenger RNA molecule to a sequence of amino acids during protein synthesis. The genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that it encodes. So, let us differentiate the translation and the transcription first in gene expression. In gene expression, transcription is the first step of gene expression where the information is passed on from one structure to another, while in translation, it is the second and last step of gene expression where the information encoded on the mRNA sequence results in an amino acid sequence. In occurs, transcription occurs before translation, while translation occurs after transcription. And in precursor, the precursor of transcription is the non-coding of antisense DNA strand, while in translation, the precursor is the mRNA produced from transcription. While in raw materials, the differentiate between the two is in transcription, the raw material of transcription is the 4 base pairs of RNA, the adenine, the guanine, the uracil, and the cytosine. While in translation, the 20 amino acids are the raw materials of translation. In initiation, the recognition of specific DNA sequences termed promoter sequences initiates the transcription that signifies the beginning of the gene, while in translation, the binding of RNA initiates the translation to the ribosomes. In elongation, the elongation of RNA sequences occurs by the binding of complementary base pairs to the new sequence, while in translation, the elongation of protein occurs by the binding of amino acids. While in product, the differentiate of the transcription and translation are: the product of transcription is the mRNA molecule, which is complementary to the DNA strand, while in translation, the product is the peptide sequences encoded from the mRNA sequence. In synthesis of transcription results in the synthesis of RNA sequences, while translation results in the synthesis of proteins. In site, transcription occurs in the nucleus of eukaryotes and in the cytoplasm of prokaryotes, where the enzymes and regulation factors are present, while in translation, it occurs in the cytoplasm of prokaryotes and in the ribosomes on the endoplasmic reticulum in eukaryotes. In enzymes, the major enzymes that are responsible for transcription are DNA-dependent RNA polymerase, while the major enzyme that is responsible for translation is amino-acetyl tRNA synthetase. In regulation, transcription is regulated by various transcriptional factors in eukaryotes and by operants in prokaryotes, while in translation, its control is mainly brought out by the binding of ribosomal units to the translation complex. In post-event modifications, post-transcriptional modifications include the editing of pre-mRNA, or the result of transcription, by the process of splicing before the mature mRNA reaches ribosomes for translation. While post-translational modifications involve the folding of polypeptide chains to obtain the three-dimensional configuration. And in detection, the process can be detected by methods like RT and PCR, DNA microarray, in situ hybridization, and northern blotting. Similarly, the process of translation can be detected by methods like Western blotting, immunoblotting, enzyme assay, protein sequencing, and etc. In inhibition, it is inhibited by some antibiotics like rifampicin and 8-hydroxycholine or hydroxyquinoline. While in translation, it is inhibited by antibiotics like tretacillin, chloramphenicol, streptomycin, erythromycin, anisomycin, cyclohexamide, and etc. So that would be all. Thank you and have a good day.